In 1928, Alexander Fleming discovered the first true antibiotic, penicillin. It was one of the greatest medical breakthroughs of the 20th century. Penicillin was considered a win in the war against infectious diseases. Penicillin saved many lives and transformed the treatment of infectious disease. Many antibiotics have been developed since then. However, the emergence of antibiotic resistance has given the bacteria the upper hand. Antibiotic resistance occurs when bacteria resist the effects of drugs designed to kill or inhibit their growth. These bacteria may infect humans, and the infections would be harder to cure. More severe infections often need extended treatment and longer hospital stays. Ultimately, healthcare costs increases. So, how does antibiotic resistance happen? To survive, bacteria can develop defense strategies against antibiotics. They carry random mutations in their genes that gets activated when they are exposed to antibiotics. The mutations cause changes in their biology, making them resistant to the effects of antibiotic drugs. As a result, these resistant bacteria can survive and reproduce even when exposed to the drug. With continued use of antibiotics, the proportion of resistant bacteria increases, leading to the emergence of antibiotic-resistant strains. Bacteria can also share their resistance mechanisms with other bacteria that have not been exposed to antibiotics. This quickens the spread of resistance. Bacteria employ various mechanisms to resist antibiotics, such as reducing drug uptake, utilizing efflux pump, inactivating a drug, and modifying a drug target. Let's take a closer look at what happens within the bacteria. For an antibiotic to be effective, it must reach its specific bacterial targets at a concentration that is high enough. Reducing antibiotic uptake allows bacteria to resist the effects of antibiotics. Some bacteria can modify the composition of their membrane, making it difficult for antibiotics to pass through. Less antibiotic gets into the bacterium, making the treatment less effective. For instance, the gram-positive bacteria Staphylococcus aureus develop resistance to vancomycin by producing a thickened cell wall that makes it difficult for the drug to enter. Bacteria have efflux pumps that sit in the membrane or cell walls. These efflux pumps can transport various compounds, including signal molecules and nutrients. Some can also support antibiotics. Mutations in the bacteria allows the bacteria to produce more of a certain pump, which can effectively remove antibiotics from the cell before they can exert their antibiotic effects. Some Pseudomonas aeruginosa bacteria can produce pumps to get rid of various antibiotics, including beta-lactams, chloramphenicol, fluoroquinolones, and trimethoprim. Bacteria can also make antibiotics ineffective by destroying or changing their chemical structure. Klebsiella pneumoniae bacteria produce enzymes called carbapinamases, which break down carbapenem drugs and most other beta-lactam drugs. Antibiotics single out and destroy specific targets of a bacterium, for instance, cell wall synthesis, protein synthesis, or DNA replication and repair. Some bacteria can change the composition or structure of a target within the bacterium, so the drug can no longer bind to the target and do its job. For example, fluoroquinolone antibiotics interrupt DNA replication. Mutations cause changes in the structure of DNA gyrase, removing the ability of fluoroquinolone antibiotic to bind to these components and exert its action. Other bacteria develop new proteins that avoid using the antibiotic's target. This is how some Staphylococcus aureus bacteria bypass the drug's effects of trimethoprim. It is clear that bacteria are very capable of protecting themselves from toxic substances to survive. We've looked at 4 established mechanisms of antibacterial resistance. There might be others yet to be understood. Understanding the mechanisms is crucial. The more we understand, the better equipped we are to combat the threat of antibiotic resistance.